Hi, everybody, and welcome. My name is Brooke Christensen, and I'm a PhD student in the Daily Lab in the Department of Ecology and Evolutionary Biology at the University of California, Irvine. I'm happy to welcome you to another in a series of workshops on integrative organismal modeling of movement. These workshops have been sponsored by the NSF Division of Organismal Systems, really with the goal of bringing together scientists across disciplines at the intersection of organismal biology and physics. So we really aim to highlight the use of model-based and mathematical tools to address fundamental questions in organismal biology. And just as a reminder, these workshops, like always, will be recorded and shared as an online resource hosted through the UCI Center for Integrative Movement Sciences with the School of Biological Sciences. And with that, I'm going to pass it off to today's workshop speakers, Dr. David Sloboda from our very own UCI Irvine, and Dr. Marion Porter from Florida Atlantic University. Thank you.
Okay, cool. Thank you, Brooke. I'm going to go ahead and share my screen. So hopefully you can see a introduction slide and a mouse moving around.
Looks good.
Great. Just organize my windows a little bit. Okay, so thanks very much, everybody, for coming. Hi to everybody who's tuning into a recorded lecture. So this is going to be the first of two workshops from our group focusing on testing the material properties of soft biological structures. So pretty much any introductory engineering or biomechanics textbook that you pick up is going to have a section on how to use stress and strain data to characterize the material properties of a structure. But those textbook approaches are often geared towards analyzing rigid materials that don't have an obvious, or that do have an obvious initial shape and that don't usually undergo very large strains. But those qualities contrast pretty sharply with many of the softer, more compliant structures that we biologists are often interested in studying, things like tendon, muscle, bone, or I'm sorry, or cartilage, tendon, muscle, or cartilage. And so the goal of our group with these workshops is to provide a really brief overview of some of the theory and the techniques that have informed our own approaches to studying the material properties of soft biological structures. So in this first workshop, we just have two talks, one from me. I'm David Sloboda at UC Irvine. I have about a 25-30 minute presentation, and then we can have a little time for questions and discussion after that. And then Marion Porter at Florida Atlantic University is going to give her lecture, and then we can have some time for questions for her specifically, and then maybe we can open it up more broadly to a general discussion if people want to stick around and have topics to discuss. So I'll kick off my first little mini lecture here. So my entire presentation today is going to focus on one question that I think seems kind of simple on the surface, but once you kind of roll it around in your head a little bit more, requires a little bit more consideration than you might initially imagine. And that question is, what is the reference length of a soft structure? I think this is a question that pretty much any researcher that's trying to characterize the material properties of a soft material or a soft structure is inevitably going to run into. And I really like this term, reference length, but you might hear this question phrased in slightly different ways. You might hear, what is the rest length of a material or a structure? What is the initial length, or even the original length of a material? For the sake of this talk, I think you can consider all these terms synonymous, and what they're all referencing is the fact that whenever we do a strain analysis on a material, strain is equal to the change in length that's experienced by that material divided by or normalized to the initial length of that material, or expressed symbolically, delta L over L naught, change in length divided by initial length. And this L naught is what all these terms are referring to. It's the reference length, rest length, or original length of the structure that you're going to normalize any changes in shape to. And in the case of something like a metal rod, I don't think many people are going to argue with you about what the rest length or the reference length or the initial length of the material is. It's the length of the rod in regular everyday laboratory conditions, right? At kind of room temperature and not subjected to really large external forces. And so determining the strains experienced by a bunch of different rods is as simple as measuring the amount of deformation that occurs and dividing by the initial lengths of these rods that you can just measure with a tape measure. But what if the original length of the thing that you want to examine is not so obvious, right? What if you're dealing with something like a muscle, which is soft and stretchy when it's not actively contracting? How do you define a reference length or an initial length for something like this? So imagine that you've just spent an hour carefully dissecting a skeletal muscle out of something like a bullfrog or a mouse or a rat, and you want to characterize the intrinsic material stiffness of that muscle. And so you fix one end of the muscle in a little clamp, you connect the other to a little, what I've drawn here is a little force transducing servo motor that can change the length of the muscle and also measure force simultaneously. And you're ready to collect some force and length data from this, but then you go, I know I'm going to need to do a strain analysis eventually with this data. What am I going to call my reference length for this tissue? What am I going to divide all the changes in length by? Maybe you just slap this muscle up on the materials testing rig at something that looked like a pretty healthy natural length. And maybe you should just call that your initial length, but then you change the length of the muscle a little bit. The force it produces is barely altered by this, and it still looks pretty good and healthy. So how do you decide which one of these is a better initial length? And maybe more importantly, when you come back tomorrow or next week and you try to run this same experiment on another muscle from a different individual, maybe one that's slightly larger or slightly smaller, how can you be sure that the reference lengths that you're choosing are comparable across those different individuals? So I'm going to present three approaches during this talk for ways to determine a reference length for a material. The first one I'll cover is an instrumentation-based approach, which really just boils down to a force or a stress-based approach. I'll talk about an anatomy-based approach, and then finally a physiology-based approach. And I'm going to use muscle as an example kind of throughout the talk. I'm a muscle physiologist by training, so it'll be, using muscle as an example, but hopefully a lot of it will be applicable to the structures you study, or that, it can at least inspire you maybe to think about how to figure out reference lengths for your subject of study. Okay, so the first option of three that I'll cover is using an instrumentation-based approach. And the general thought behind this is that, well, hey, I'll put my sample up on the rig, and whatever length I can start to detect tensile force at, I'll call that my reference length. I'll define as reference length. So practically, you would attach something like a muscle to your force transducing rig and bring it down to a length where it's obviously slack, not stretched at all, not producing any tensile force. You might zero out your force transducer so that the weight of the muscle isn't being accounted for. And then you could incrementally stretch out your muscle in millimeter or sub-millimeter increments. Always give it enough time for any viscoelastic effects to settle out, and measure the force it's producing as you slowly increase the length. And you could keep increasing the length until you get a reading from your force transducer that's equal to the lowest resolvable force from your force transducer. So let's say this little servo motor can resolve a force of about 1/10 of a Newton. So when I eventually get that 1/10 of a Newton reading, the length that corresponds with that 1/10 of a Newton reading, I'll define as my reference length. And kind of the deeper rationale behind this approach is that for all the samples, the reference length that you're defining is going to correspond with a similar mechanical state, a state in which the muscle or the tendon or whatever you're studying is just starting to resist tensile deformation. So any elastic elements in the tissue are just starting to be engaged. It's kind of just barely been drawn taut. And I think this approach makes a lot of sense. Mechanically, it's pretty intuitive, and it is indeed a common method that's used in the literature to define reference length for biological materials. But it's not perfect, and I wanted to present just a couple cautions that you will want to think through if you're applying this method. The first caution is that when you use this method, the sensitivity of your transducer is ultimately going to determine how accurately you can identify when the structure starts producing tension. So if you had a transducer with higher resolution than 1/10 of a Newton, you would inevitably see that somewhere in between these two states, you started to generate tension. You just couldn't see it because your force transducer was not sensitive enough. So when you do this approach, you're not really putting your sample at the state where at the point where it starts to develop tensile force. You're putting it at a length that corresponds with this somewhat arbitrary force that is defined by the sensitivity of your motor. And practically, whether or not the error that introduces is going to be important or significant enough to affect your results is going to depend on, how sensitive your transducers are and the material properties of the material you're studying. But I think ultimately it is important to at least, be aware that when you're using this approach, it essentially boils down to a force-based approach where you've defined a force that you're going to stretch your sample to and call that your reference length. And one potential pitfall here is that there's a little bit of circularity involved in that. So the intrinsic stiffness of each of your samples, which is ultimately often the thing you're trying to measure, will influence the length you choose as a reference length. So if you had a couple muscles in your experimental set that were just kind of intrinsically more compliant than the others, those would be getting stretched to a slightly longer initial reference length to get to that 0.1 newtons of force. And then when you go and analyze your stress strain data, you might never see that those muscles were slightly more compliant because that information got washed out when you chose the reference lengths for those muscles. So one way that you can improve this method a little bit is by choosing an initial stress to stretch to rather than a force. To help account for differences in the cross-sectional area of the samples. So if you can measure or calculate the cross-sectional area of your samples, instead of putting that at kind of an arbitrary force of something like 0.1 newtons, you could put them at a stress you choose of like 1 Newton per centimeter squared of cross-sectional area and call whatever length that corresponds with your initial length. That way you would at least be determining rest length using a load that's proportional to the size of each sample. So in general, these kind of force or stress-based methods are relatively easy. They don't require a whole lot of prep work. You can perform them on samples kind of that are completely removed from their anatomical context, and they really don't take a lot of time in your experiments. But they do involve some pitfalls that can have effect on your data, mostly stemming from this kind of circularity between force and length. And that, definitely doesn't discount this method, but it's something you want to consider as you think about your particular experiment. Okay. So another approach to determining appropriate reference length for a set of soft structures would be to use an anatomy-based approach. And so the general idea behind this is that all of these samples came from somewhere. They had some original anatomical context. So why not use the length of the structure in the actual organism, the in vivo length, to define a reference length? For example, if you had an anesthetized or a recently euthanized bullfrog, you could look at the muscle of interest before it's dissected out of the organism, see how long it is, and use that as your reference length. So this is going to be, I think, a little bit less precise than using a force or strain-based, excuse me, than using a force or strain-based approach, because you don't have that hyper fine control of length that's going to allow you to get lengths corresponding with kind of 0.1 Newton differences. But it is really nice in that this is approach would kind of completely avoid that circularity I touched on in the last slide. It provides an independent metric of rest length. And kind of the rationale for this approach would be that for all samples, the reference length will correspond with the length of the structure in a defined anatomical position. And I think this is an attractive method because it seems very straightforward, but the biggest challenge here is that it's going to introduce a lot of variables that are going to be important to consider. And so there's just going to be more chance for confounding details to slip into your data. So some things you would really want to consider before trying an approach like this is you want to first and foremost think about how you will produce a reference position in every experimental animal. So changing the orientation of the joints, moving the limbs around is of course going to deform all of these soft structures inside the animal. So you need to think about how you're going to define and maintain a reference position for this limb. You could do something like define an angle of the thigh relative to the trunk and of the shank relative to the thigh. Maybe even use some constraints to put the joints into that conformation every time you do a rest length. You'd also want to, consider how you're going to account for the 3D complexity of the structure you're interested in. So biological structures don't often play nicely in 2D planes. So for example, this sartorius muscle that we're looking at from this bullfrog could be kind of bending up out of the page at us in a way that's not represented by this 2D illustration and wouldn't be represented by something like a photograph. And so you'd want to account for that by, as simply as using a flexible tape measure to try to get the actual path length of the muscle, or something as complex as taking a CT scan and really measuring the path length of the structure you're interested in 3 dimensions. And finally, something that seems kind of obvious, but it is very tricky, is you'd want to really establish how you're going to define the endpoints of the structure. How are you going to define where you mark your origin and your insertion for the sample? Biological materials often merge into other structures or have complex attachments. And so you might want to do some dissections and really establish how you're going to define the endpoints of your structure so that you can be consistent from sample to sample. And finally, I want to caution that there is kind of an inherent assumption that's being made if you're going to use this reference position idea. And the assumption is that the reference position chosen puts the relevant structures in kind of the same physiological state across all individuals. Kind of for lack of better words, it's assuming that it feels the same for 2 organisms. So imagine if I sit cross-legged on the floor and you sit cross-legged on the floor so that our limbs are in anatomically similar positions. Who's to say that the muscles are in physiologically similar states, right? So maybe you stretch a ton and your muscles are used to being at those lengths, whereas I don't. Alternatively, maybe our personal anatomies are just different enough that our muscles are put into pretty different conformations, even though we're occupying postures that on the surface look similar. So both the lifestyle of the organism and its personal anatomy are going to affect the soft tissues inside of it in any given position. So the fact that uncertainty or that assumption is there doesn't necessarily torpedo this approach. Maybe that's exactly what you're interested in studying, maybe differences in the stiffnesses of the tissues across joint angles or something like that. But before, using this as your primary method of defining a reference length, you would want to make sure that this is a method that you can rely on. You might go in and do a separate mini study where you look at something like sarcomere lengths in the muscle at in the reference position that you're interested in. And if you could show that it was consistent in the position you've chosen, then maybe in future studies, you could rely on reference position a little bit more heavily as a way to establish a reference length. So overall, it's a method that I think seems kind of simple and obvious on the surface, but you would really need to think about kind of these considerations. And you'd want to think about ways to ensure that your reference anatomical position actually puts your structures of interest in kind of a comparable state, in a comparable state that makes sense for the question that you're interested in asking. Okay, and so the last approach I'm going to highlight is one that is somewhat specific to muscle, but maybe it'll get non-muscle physiologists thinking about ways to apply a similar strategy in their own areas of interest. So the general idea would be that you use some intrinsic physiological behavior of your tissue to identify a common reference length. And the example that I'm going to give is a technique that's common in the study of muscle mechanics, where we use the active contractile properties of muscle to define a reference length. So muscles exhibit this really fundamental behavior where the contractile force they produce varies as a function of the length that they're operating at. So in other words, all muscles have kind of a sweet spot length where the contractile force they produce is maximized, and operating at either longer lengths or shorter lengths decreases the maximum contractile force they produce. And this ultimately stems from the fact that muscles produce force using contractile proteins that need to overlap each other to some degree and tug on each other. And if you change the length of the muscle at a gross kind of macroscopic scale, that changes the amount of overlap between the contractile proteins. So let's say we have a muscle again connected to a force transducing servomotor. We put the muscle on our rig so that's kind of just barely taut, and we stimulate the muscle to contract with a pulse of electricity. In this made up example, we've got a muscle at 70 millimeters length, and it produces 8 newtons of force when we stimulate it to contract. If we then increase the length of the muscle a little bit to a new kind of static length, and we stimulate it to contract again, we would see that it produces a different amount of force, this time a little higher. And we can repeat that process over and over again. And if we do this and explore a wide enough range of muscle lengths, we'll end up with this very characteristic curve that emerges. That is a curve that pretty much all muscles are going to generate with this general shape. And what it reveals is the range of lengths over which the muscle produces maximum contractile force. And generally what we would do in the field of muscle mechanics is we just fit a polynomial equation to this curve, interpolate between the points, and identify the length at which maximum contractile force is produced. And we would define that length, the length at which maximum force is produced, as L naught, so as our reference length. So in this made up example, we've got a reference length of 72.2 millimeters corresponding with maximum force produced. And the rationale, kind of deeper rationale behind this, is that for all samples, our reference length that we define will correspond with the physiological condition that is ideal for force production. So the contractile proteins will all be overlapping to a similar degree. In that way, kind of no matter how large the muscle is that we pull out of our different experimental animals, when we go to set them up on our stress strain curves, we can kind of shuffle them all together and line them up based on the force that they produce maximum, the length that they produce maximum contractile force at, using that as our reference length. So this is kind of a standard approach in the field. It's nice in that, just like the anatomy-based approach, it provides kind of an independent way to determine a reference length that's not influenced really by the intrinsic material properties of the sample. But like the other ones, there's some important considerations and things to keep in mind if you want to try this. The most obvious one is that it requires live, healthy tissue. Any issues with your muscle are going to influence the active length tension curve and influence how you can define this reference length. It requires a lot of extra work. So it requires an extra data collection process. So collecting this curve can take an hour or two once you get the muscle all set up and actually do all these contractions. And time can be a kind of a precious resource when you're dealing with live biological materials. It also requires extra equipment. In this case, a rig for stimulating the muscle to contract actively. And it requires some extra planning. So when you do experiments like this, you have to figure out how you're going to stimulate the muscle using either a nerve cuff or using field stimulation. And those kind of considerations can really limit the muscles that you actually have access to, the muscles that are going to work for this setup. And then finally, there's a lot of skill and practice involved, right? Your ability to measure this active length tension curve accurately will determine, how confident you can be in your reference length that you're choosing for each specimen. Okay, so now I'm going to wrap up with just some general conclusions about this kind of initial question I posed. What is a reference length of a soft structure? So overall, choosing a reference length is all about finding a way to put your samples into comparable physiological and mechanical states. And it's important to recognize that there is no inherently correct reference length. That's why I like calling it a reference length rather than an original length or a rest length or initial length. Because those things don't really exist for a soft structure. This whole idea of a reference length is something that we've conceived to help with our stress strain analyses. So there is no true answer. And so what that means is that you as a researcher get to define reference length based on what makes sense for your particular question and your particular approach. Often there will be established ways that people in different fields establish a reference length for a particular structure. You'll want to make sure you read the literature associated with your material of interest and see if you agree with how it's done and use that method. Or if you choose a different method, just be ready to defend your choice to reviewers and to colleagues. But it is up to you ultimately to determine what reference length you choose. And so I presented a couple different examples of how to do this and presented a lot of caveats, and it can be kind of overwhelming. But my advice would be, if you're going to collect data and you don't know which reference length is going to be best for you, one solution is to take many measurements that you can use later to reanalyze your data. These strain calculations are something we often do after data collection. So collect all the information that you can so that you can try a couple different reference lengths later. For example, you'll want to always measure cross-sectional area or measure some combination of mass, volume, and density that allows you to calculate cross-sectional area later and define maybe a stress-based reference length, which can always be used kind of in a pinch. You could measure an anatomical reference length kind of as you go through your normal dissection, so that you just kind of have it in your lab notebook there and you can use it to check why some large outlier, try to understand why it's there. It's handy to always have this reference length. And then once you have your sample on the rig, you can always do a quick measurement of the length at which force starts to be produced, like the first force or stress-based approach, which can also be used as a reference length if you need it. So try to collect all of this information. With all of it in hand, you can always reanalyze your force length data after you collect it, to see if what makes sense for your data, and to make sure that you're not missing some interesting part of the story that you're exploring. So thanks very much. That's all for me. I'd be happy to discuss other people's approaches to this, or answer any questions if there are any. So thanks very much.
Hey, thanks, David. That was great. People can feel free to use the Q&A function or the raise hand function to ask any questions. We can also wait till the end after Marianne's session and do them as one dedicated Q&A. Thanks, everyone.
So Liz Mendoza asked, how would you go about finding the reference length for a material like tendon or aponeurosis? I think, thanks for the question, Liz. I think either of the first two approaches would be appropriate for that, either defining, an initial force that you're going to stretch to, and whatever length corresponds with that force, you could define as your reference length, or defining a stress that you're going to go to. or looking at the anatomical context and looking at maybe the length of the tendon you're interested in the actual biological organism. But yeah, thanks for the question.
Great. So I'm going to follow up a little bit. We've learned how to find our reference length. Now I'm going to talk a little bit about how to do some testing. I study mineralized cartilage, so we're going to be using those vertebrae as an example for how to do this. And major players that we'll be talking about here, we can see in this vertebral column, we've got vertebrae, joints, and more vertebrae. And this is all mineralized cartilage in the vertebrae. So we're talking about something that is stiff, but it's also viscoelastic. So we're going to be talking about a number of approaches to talk about and think about studying these viscoelastic materials. And more specifically, I'm going to tell you a little bit about some of the quasi-static testing I've been doing, varying strain, some dynamic testing, and then a little bit about the nonlinear dynamic mechanics. So the dynamic testing and the nonlinear dynamic mechanics, of course, we're talking about cycling our testing. I did it in bending, but anytime you're looking at cyclical testing, this would probably work for you. And so when we're thinking about viscoelastic materials, we're thinking about materials where strain and strain rate will be important. So first off, we'll talk about the quasi-static test. And what I was thinking about in these experiments was exploring the quasi-static mechanics, just to give me a sort of broad idea of a way to characterize these materials, and looking at them at relevant strains. And I wanted to do this at biologically relevant strains, which means I'm going to be working everywhere in the elastic region, so before the yield point. We'll see a picture with this. And this was really all inspired by my testing to failure. So I was testing these vertebrae and breaking them. And like I said, quasi-static testing is a great way to give you a baseline understanding of how a material is working. And this testing to failure really let me know sort of what the upper limits that this material could do. But that's not super biologically relevant because most organisms are not going to be commonly loading any of their materials to fail. So this is where I was really inspired by the yield point, actually. So here's just an example of a stress strain curve from one of these mineralized cartilaginous vertebrae. And we have strain on the x-axis. We just talked quite a bit about that reference length, and then stress on, or strain on the x-axis, stress on the y-axis. And some of the things we'll be talking about today, just we can measure stiffness, this linear region, and strength is where our material is going to break. But this yield point in my mineralized cartilage, I get this really clear yield point. And this is where the material is going to transition from the elastic to the plastic phase. And this means beyond the yield point, the material is going to permanently deform. And in the mineralized cartilage I study, we do see this very dramatic yield point. And this led me to start thinking about what happens before yield, because like I said, organisms aren't going to be breaking their materials just to move. That wouldn't be a particularly useful strategy, I would imagine, for most things. And so I really began to look at what's happening pre-yield. And because we are looking at viscoelastic materials, we can think about the strain rate, so how fast we're testing the materials. And then we can also vary how much strain we put on the materials in this pre-yield zone. And you can see that in this particular example, we're getting about 12% strain, maybe 10% strain. So we could be working in this area here to start thinking about what is happening. And if you look at the material I study itself, it can be rather there's a lot going on there. I don't want to say too complex, but we do have all these plates. There's a lot of structure there that is creating this viscoelastic material. So we've got this mineral, but it is cartilage. So there's a lot happening here. And I was really interested in particular, how these different pieces of my structure are impacting the mechanical properties. And for simple quasi-static tests, we can just do mechanical testing where we put our vertebrae on the platen. We've got an actuator that's going to move down and apply some forces. And we can either break our material and test it to failure, or we can test it at strains within that elastic zone. So this is an example of when we did that. So we were able to really focus. In particular, I was interested in about 4% strain because that was values that I had measured in vivo. And so I was really wondering what was happening to the vertebrae and how they were working there. So what we were able to do is start working and not testing all the way out here to failure, but just working within in this sort of pre-yield zone. Because yield point was really interesting to me, that was a good point. But then there are also practical considerations. So my system that I have only has 1,000 Newton load cells. So I also can't fail these materials, generally speaking. So I was trying to work within interesting biological questions, but also this is just practically useful if you're constrained by like the size of your load cell. You know, you can learn a lot, like I said, from the quasi-static testing and breaking your material, testing to failure, but also testing within these lower strains can be really useful. And so again, like I said, one of the things we can do is we can vary how much we strain our materials. And because we are dealing with viscoelastic materials, how much we strain and our strain rate during testing is going to impact the results we get. And we can learn about our materials by doing so. So, you know, here in this example, we see that 10% strain, we're going to get stiffer and tougher stress strain curves. And again, we can start to think about our biological systems and put that in the context. But this was one of the things that I thought really solved a problem that we were trying to get at in our lab, but it also was a pretty simple solution to starting to look at how this material worked in a pretty simple way with that quasi-static testing. The next thing I wanted to talk about was some dynamic testing. So here we're just really focusing on bending our vertebral column in this example. So we're going to be just bending that column and thinking about the intervertebral joint. And we were doing this in the context of freezing, impacting the mechanical properties of those joints. So especially once you start looking at things like our vertebrae, where we had that mineralized structure, now we're building out that structure to look at a vertebral column. This dynamic testing can be really useful anytime you're looking at cyclical data. So you could do that even with of course, the muscles we were just talking about. So we're thinking about comparing these properties during bending. And here's a picture of those joints I was interested in. So they're going to be these fluid-filled capsules in the vertebral columns of these animals. And the anatomy was going to vary between the species. So that's why it was really interesting for me to explore this because the squishy bits often get dissected away when I'm looking at the vertebral columns. So here is our setup. What we were able to do is we were able to take a linear system. So this is a material testing system from MTS, and we've got our force transducer, our load cell right here, and our actuator right here. And this was a linear system that kind of moved in this direction, if you can see my arrow. And what we did was we were able to modify this so that we were able to look at bending And we did that by applying couples here at the end. So you can see we've got this little moment arm attached to our actuator and our load cell, moment arm coming down. We've got a little joint right here, little hinge, and we can change the height of these grips so that we're able to change the length of our moment arm, which is going to, of course, allow us to test another variable and look at how bending changes in our structure. So here's our reference structure, just a little piece of rubber that's held in these clamps. And one of the things we're able to do is we're basically able to move these two joints. They come closer together and our structure is going to bend. And we can use this setup now to test different frequencies. We tested a whole sweep of frequencies. We can test displacement by varying the actuator, the displacement the machine does. And we can also change the moment arm and think about how that's going to impact our bending. And then one of the other things we were able to do is we were able to, because we were using biological materials, we were able to put this in an environmental chamber with Ringer solution to start making sure we were keeping our tissue hydrated. That's something else David also touched on was, what happens in these tissues inside versus outside of the body. And anytime, we dissect, especially these squishier viscoelastic materials, they're going to start to dry out, and that can become a problem, or you can have your muscle start to die. We were able to do this. We calculated, I'll just show a few things what we looked like, what our data looked like. We have some frequency sweeps. So we're looking at a number of curvatures produced by our system. This is just at one frequency, and we're looking at the structural stiffness of our different treatments here. And we can see how that changes over a range of curvatures. Not too much change here in this example. So here's when we were calculating work to bend. We start to see that there's a clear curvature trend in our data. So how we're bending it, how we're bending our material is really going to impact the values we're getting. And again, depending on what biological system you're putting this material into, you can think about what that means. Because I study vertebral columns, thinking about curvature related to swimming is sort of the biological context for these data. The last thing is where I wanted to spend a little bit more time. And this is thinking about nonlinear dynamic mechanics. So this was something I had to spend a lot of time learning, and I was able to work with an engineer. So in this particular example, we're thinking about nonlinear viscoelastic properties. And I was thinking about, again, because we're talking about dynamic mechanics, changing with relation to frequency of our testing. And again, thinking about putting this in the biological context of a fish, I was thinking about swimming speed, how fast animals are swimming. And then also the amplitudes that we're testing in. So again, thinking about that displacement. And again, that's something related to specifically how my system moves. So you'd have to think about your system. Are we talking about muscles, how they're moving? Are we talking about wings? Putting your work in that biological context, your material. So for these fish, that's what we were really thinking about. Here's how these nonlinear properties work. This is sort of a biologist explanation of engineering. So again, we're thinking about stress and strain. When we think about linear properties, we are thinking about examining differences between our cycles. So if we are testing a muscle, we can think about cycle 1, cycle 2, cycle 3. If we're thinking about testing my vertebral column from a fish, we could think about bend 1, bend 2, bend 3 as the fish would be wiggling its vertebral column to swim. And when we get these linear properties, we can calculate things like E prime and E double prime. So E prime's the storage modulus. So we're looking at the slope of the major axis here. And this is going to be the midline of our work loop. And then we've got the loss modulus, which is the minor ellipse here. If we're thinking about nonlinear properties, we're going to be thinking about examining between and within cycle differences. So not only what's happening between cycle 1 and cycle 2, but what's happening at the beginning of cycle 1 and at the end of cycle 1. And so we're thinking about reconstructing this midline like we did in our linear mechanics, and it's not quite going to work as well. It won't be a straight line. It's going to be curved. And one of the useful things about thinking about these nonlinear properties is we're going to get a little bit more. We might lose some information if we're assuming this straight curve, these linear properties. We might lose some of the complexity of what's really happening. So for example, we can measure E sub L, which is our elastic modulus at the largest strain. So this is looking at our modulus over here where we would have large strain. So we've got strain on the x-axis here and stress on the y-axis. And here's one of our cycles. The little gray arrows indicate the direction of the cyclical testing. So we can measure stiffness at this largest strain. We can also measure stiffness here at this minimum strain. And you can see that this work loop, it reminds me of a C. elegans actually, but these can take a variety of shapes depending on the material you're looking at. And one of the things that I was particularly interested in was stiffness, because that's something I looked at in the structural stiffness for that dynamic bending, and then in my quasi-static properties. So that was one of the things I was very interested in. And one of the things we can measure is an index of nonlinearity. So we can think of the relationship of the elastic modulus at largest and minimum strain and start to think about whether our properties are linear or nonlinear. And the way this setup works is very similar to my last setup. It's just a matter of how you analyze your data, right? So doing some extra tricks with our work curves, our loops that we're producing. So again, we're able to use our force transducer and our actuator, and we can measure a variety of displacements, frequencies, and the length of the moment arms. And we can measure a whole suite of variables and viscous properties as well. I don't want to go over all of those because it would be a very long talk and you're probably wanting to get lunch or something, I don't know. So here we're looking at some of our linear viscoelastic data. So we've got force and displacement. Force is in this pink color and displacement is in blue. And so we can see that we have very linear data, differences between cycles of varying frequencies and amplitudes. There's not a lot of offset here, for example. When we're looking at our nonlinear properties and calculating it within this framework, we get a lot more information. And so this was something we were able to modify from work that Randy Ewalt did during his dissertation. And he was looking at low amplitude oscillatory stress. He's a rheologist, and we wanted to put this in the context of bending. So we called this low amplitude oscillatory bending as we were starting to look at this. And so if we look at this center curve here, our blue line is our filtered work loop, and the red line is going to be the interpolated midline of that work loop. And this is going to be from the line from which we're going to be measuring our properties. And when we look at this, again, we sort of see that C. elegans shape in this particular curve. And if we were just looking at E prime, that storage modulus, we would assume that this is nice and straight. So we make that assumption in our data analysis. Whereas if we look at the slope at the largest strain and the minimum strain, we're going to see something a little bit different. And so this was for our stiffness values or our elastic moduli variables. This panel right here, this is for our viscosity measurements that I'll show you some examples of those. And we can see that, again, for our viscous properties, this is very nonlinear as well. So we don't have nice straight interpolated line using this framework. And this is a code from MATLAB that Randy had developed. So if we just look at what a lot of my curves just look like after doing experiments, if we look at increasing frequency along the x-axis and increasing displacement along the y-axis, and then each of these curves, we've got force and displacement. So very similar to what you see. Think of this as just a big matrix. Sometimes we have curves that look more linear. They're nice and kind of circular. But then sometimes we get these non-linear, again, I think of them as kind of C. elegans curves. And then over here in the red, we actually start to see some sort of capping off. We see it down here in the blue at the ends where we get that maximum strain and the least strain. But these diagrams are really useful because they're going to give us an idea of what our loops, what our cycles look like over the range of the variables we're looking at. And in these particular examples from the mineralized cartilage, we were getting higher displacements or getting more non-linearity over here. So at higher displacements and higher frequencies in this particular example. And one of the things I want to show you is an example of just our stiffness at largest strain and minimum strain. And what we found was that in the case of the largest strain, we got increases related to curvature and frequency with our statistics, and stiffness at the largest strain was similar to our storage modulus. And when we were looking at stiffness at minimum strain, we saw increases with curvature, but not with frequency. So you can see here, we've got curvature on the x-axis and our frequencies are the different colors of the lines and the dots. And there's not a lot of difference in the slope of these lines. So they were not significantly different. So what we get out of our data could be very different, again, depending on if we're looking at stiffness at the largest strain or at the minimum strain. And in our particular case, for these vertebral columns, our largest strain stiffness was similar to our storage moduli that we calculated. And so what this can tell us is that when we're looking at our linear data from a linear standpoint, we're going to have our storage modulus be greater than our loss modulus. So our system is going to be dominated by those elastic properties. And we did find that the storage modulus and the largest strain, stiffness at largest strain, are statistically similar. But our linear measurements ended up actually overestimating the stiffness of the cycle because they didn't take into account the stiffness at minimum strain. So that's one of the powers of this framework and this methodology, is it does allow us to look in more depth at what's going on there. And so here's examples of this. Here's our storage modulus again, and our modulus at least or largest strain. And what we ended up finding in terms of the elastic properties is many of the properties, both the linear and nonlinear, ended up being frequency and amplitude dependent. So how you're setting up your test, thinking about those frequencies, the amplitudes of your test. And again, we also varied the moment arm of our bending those are going to really impact our results as we look at them. And then sort of thinking about it in the context of that biological context. So it's not just the material, it comes from an animal. And so what we found is our vertebral column is going to stiffen under certain testing parameters and result in a stiffer spring that would probably result in faster swimming. Another way that we can use this framework is we can look at the viscous properties. So here we're going to have viscosity at largest strain and minimum strain. And again, we can see that at largest strain, we're going to be seeing significant differences with curvature and frequency. And we're going to see that our viscosity at minimum strain, we're going to see increases with curvature. So our lines are pointing up, but actually we saw decreases with frequency. So our faster frequencies, these warm colors, are actually lower in our viscosity than they were in our elastic test. And so what we were able to look at this in the end was our linear viscous forces are going to dominate the low frequencies and larger curvatures, and the viscous forces are going to be greater when strain is minimum. So again, we were able to learn an awful lot about our system by parsing it into these linear and nonlinear effects. And when we look at that index of stiffness at largest strain to minimum strain, one of the things we can see is this reference point. So if we're seeing changes in curvature and frequency, we're going to be above 1 if the spring is stiffening within the cycle, and below 1 if our spring is softening. So what this all means, we've been able to test with a variety of different mechanisms to start to really explore the viscoelastic properties of this mineralized cartilage. And so this is important because it allows us to think about the dynamic properties in a physiologically relevant set of mechanical behaviors. The nonlinear properties were really great because they allowed us to understand the variability of the mechanical behavior. So again, thinking about what's happening at the minimum strain and the least strain. And then again, it allows us to put our work back into the animal and think about that biological context. So with just our dynamic testing, we can get those really great measurements of the storage and the loss modulus. With the nonlinear framework, we can get strain at largest, or we can get values at largest strain and minimum strain. And then with that quasi-static testing, that gives us a really good broad picture of how a material is going to work. And as we start to explore the quasi-static behavior, just varying the strain rate, how fast we're running our test, and how far we strain the material are all going to give us really great insights into overall how our sort of squishy biological materials are going to work. And with that, I will see if there are any questions.
Looks like we have one question in the Q&A.
For testing material properties, are you also able to adjust the unloading rate or the material? I think it would really depend on the system you were using. My system, yeah, I mean, I think you could adjust how fast you were running it, running your test. So for the quasi-static test, you could release the material slower. So you could have a particular loading rate, and you could change your unloading rate to be slower or faster, depending on what you wanted to do. Just anytime you're working with those viscoelastic materials, remember the classic like Vogel example of silly putty, right? If you pull a silly putty really hard, it's going to snap. If you let it ooze or pull it slowly, it's going to kind of ooze. So yeah, if you were doing quasi-static tests, sort of how you're releasing the platens or putting the clamps back together if you're testing in tension. In my bending setup, I think it would be a little bit harder because I'd have to think about how my hinges were working on my my little setup in loading and unloading if I was going to do that at differential speeds, like different speeds. So, but I think it could easily work in quasi-static testing.
If nobody has any other questions, I do want to make a note that I dropped the research web pages of Marianne and David in the chat function if you want to learn more about their research and how to contact them with any follow-up questions. And just one more time, the workshop recording will be posted on the SIMS website, and we will have part 2 of this series on Friday, June 17th from 10 to 12 p.m., which will be a further elaboration of the discussion on how to test these soft biological structures. Thank you guys for being here.
Yeah, thank you for organizing, Brooke. And anyone who wants to chat about their experiments, either on live or on the recording, definitely just email me. Always happy to chat.
Same.
Okay, with that, we're going to go ahead and wrap it up and head out. Thanks, everyone.